So let's kind of review everything that we've talked about so far with action potentials. So let's look at what the main events or the main phases of an action potential are. All right, so we have that depolarization phase of the action potential that's caused by the opening of voltage-gated sodium channels. Sodium rushes in, that causes an increase in the transmembrane potential, and that's important because that increase in voltage is going to cause the release of neurotransmitter by the end of the axon. Then we have a repolarization phase of the action potential, and that's caused by the opening of voltage-gated potassium channels. Right? As the potassium leaves the neuron, the transmembrane potential decreases, going back closer to resting potential. Now don't forget, those voltage-gated potassium channels kind of suck at closing, and we hyperpolarize a little bit at the end of the action potential. So we go a little bit beyond resting membrane potential. Okay, so let's take a look at this graph. At the bottom, we can see time, and on the left-hand side, you can see voltage. So voltage is increasing as we go up. So right now, we're starting off at resting membrane potential. We're at negative 70 millivolts. At negative 55 millivolts, we open our first voltage-gated sodium channel. Right? So negative 55 is that threshold point to open our first voltage-gated sodium channel. Now don't forget, that's knocking down our first domino. So if we open our first voltage-gated sodium channel, then we will open all of the voltage-gated sodium channels all along the axon, right? And we're going to get a big change in voltage, big dramatic change in voltage up to around plus 35 millivolts. These voltage-gated sodium channels have a timed closure, and they close automatically after 1 millisecond, and that happens to also be around plus 35 millivolts. Now that plus 35 millivolts is significant because it's the voltage that we need to achieve in order to open our voltage-gated potassium ion channels, right? So at plus 35 millivolts, we're going to open our first voltage-gated potassium ion channel. And of course, these are also like dominoes. Once you knock down the first one, all of the rest of them will open, and we get another large change in voltage, but this time it's a decreasing change in voltage, bringing us back closer to resting potential. This is our repolarization phase of the action potential. We're repolarizing, getting closer to resting potential. Now the voltage-gated potassium channels suck at closing. They begin to close at around negative 70 millivolts, but they don't finish closing until we get to about negative 90 millivolts, causing a little piece of hyperpolarization at the end of the action potential. Now at this point, we're done with the action potential, but we still have further voltage changes that occur. We need to get from negative 90 back to resting potential of negative 70. And this is where the sodium-potassium exchange pumps are really important. Again, this is not part of the action potential. This is something that's happening after the action potential is complete. We're returning to resting membrane potential. And the way we do this, the way we get from negative 90 to negative 70, is to use the sodium-potassium exchange pumps. And they're going to put the sodium and the potassium back into the correct positions. Because don't forget, at the end of the action potential, we move sodium inside and we move potassium outside. But that's not where we want them to be, right? We need them to switch around. We need the sodium to go back outside, and we need the potassium to come back inside. But we have to use the sodium-potassium exchange pumps to put the sodium and potassium back into the correct position. While we're moving the sodium and potassium back into their correct positions, we're also going to correct that voltage number back to negative 70, right? So we'll fix everything by using these sodium-potassium exchange pumps. And then we're back to negative 70, which is our resting membrane potential. So this is all of those changes that we just saw put together on one graph. Now here, what I've done is I've taken out the pieces of the graph that were not the action potential. So we're looking at only the action potential portion of the graph, right? So don't forget, the action potential is that wave of depolarization followed by the wave of repolarization, and then a tiny little bit of hyperpolarization there at the end. Those are the main parts to the action potential. Okay. And this graph is just kind of emphasizing the difference between resting potential, action potential, and then that return to resting potential. So this little blip right here at the end is moving basically back to resting potential once our action potential is complete. And this little blip here is significant, this return to resting potential, because that's where those sodium-potassium exchange pumps are doing almost all of their work. Now here in this picture, what we're doing is we're kind of teaming up a picture of the status of the voltage-gated ion channels or sodium-potassium exchange pump with the different parts of the graph that we were just looking at. For example, at resting potential and return to rest, we're highly utilizing our sodium-potassium exchange pumps. Those sodium-potassium exchange pumps are actively transporting sodium and potassium against their concentration gradient, right? They're moving the potassium to the inside of the neuron, even though there's already a lot of potassium inside the neuron, and they're moving sodium to the outside of the neuron, even though there's already a lot of sodium outside the neuron. During the depolarization phase of the action potential, so this part where we're moving from -70 to plus 35 millivolts, that's the depolarization phase, we're going to be highly utilizing our voltage-gated sodium channels. The voltage-gated sodium channels will open, sodium rushes into the neuron following its diffusion gradient, causing the voltage to increase inside the neuron through the addition of those positive sodium ions. The repolarization phase, moving from plus 35 down towards -70, is caused by the opening of voltage-gated potassium channels. When we open those voltage-gated potassium channels, potassium diffuses out of the neuron, and as positive ions leave the neuron becomes more negative inside, getting closer to that resting potential. So an overall summary of an action potential. An action potential starts when the axon hillock reaches threshold of -55 millivolts. This happens through graded potentials, specifically those excitatory postsynaptic potentials. In the beginning to middle part of an action potential, we have a wave of sodium diffusing into the axon, and this begins at the axon hillock and it runs all the way down to the axon terminals. And this is that wave of depolarization that's caused by the opening of our voltage-gated sodium channels. Now don't forget, the whole point of this depolarization phase of the action potential is to cause the axon terminals to release neurotransmitter. In the middle to end portion of the action potential, we see a wave of potassium diffusing out of the axon, beginning at the axon hillock and running down to the axon terminals. This is the repolarization phase of the action potential, and it's caused by the opening of voltage-gated potassium channels. At the very end of the action potential, we see a little bit of hyperpolarization due to the slow closing of voltage-gated potassium channels. So that's it. That's the end of the action potential. But just a quick reminder that once the action potential is complete, we're at -90 millivolts and we need to be at -70, and we get to that -70 point by the use of our sodium-potassium exchange pumps. So let's kind of review these transmembrane potential changes again, just in sort of a different way, okay? So let's start at the very beginning. How do we get that initial change in voltage to start the action potential? Well, it all starts with neurotransmitters, right? Neurotransmitters open chemically gated sodium channels, causing excitatory postsynaptic potentials at the dendrites or cell body of the neuron. Right? If enough sodium enters the neuron during these excitatory postsynaptic potentials and we reach -55, then we will open our first voltage-gated sodium channel, right? And that's the beginning of the action potentials, the opening of the first voltage-gated sodium channel. That's the knocking down of our first domino. If we knock down our first domino, all the rest of them will fall down. So that's when we move into the action potential. We get all of the voltage-gated sodium channels opening all along the axon. That's our depolarization phase, followed by all of the voltage-gated potassium channels opening all along the axon, and that's our repolarization phase. And again, just to remind you, the whole point of the action potential is to cause a release of neurotransmitters at the axon terminals, And we talk a lot more about that in our synaptic transmission, or if you will, synaptic activity section. Before we leave action potentials altogether, I want to talk about propagation of an action potential. So to propagate an action potential is to move an action potential. We already know that action potentials move and they travel. And we know that action potentials are capable of traveling through that domino effect like mechanism of opening all of those voltage-gated sodium channels. But it turns out that propagation can actually happen in two different ways, depending on the type of axon we're using. So there are two types of axons that we can use. A myelinated axon, and that's an axon that has those myelin sheaths on them that we talked about way in the beginning of our neural activity section. And then we have axons that are unmyelinated. They have no myelin sheath around them. These two different types of propagation are referred to as continuous and saltatory propagation. Continuous propagation is what occurs along an unmyelinated axon, and it's not very common because unmyelinated axons are not very common. Most of the propagation that occurs in the human body occurs through saltatory propagation because this is the type of propagation that occurs on the more common myelinated axon types. So let's take a look at these two types of propagation. We'll start with the continuous propagation because this one's a little bit easier and it's more akin to how we've been talking about action potential movement all along so far. With continuous propagation along an unmyelinated axon, what we're seeing is that every single voltage-gated ion channel along the whole entire axon has to open. Okay? So we're opening every single voltage-gated ion channel. We're knocking down every single domino. Right? And that's how we've been talking about action potentials so far. Turns out, though, this is a pretty slow way to move an action potential, and it's also very energetically costly. If we open every voltage-gated sodium and every voltage-gated potassium ion channel all along the axon, we will displace a lot of sodium and potassium, meaning we have to replace the sodium and potassium back into their correct positions. And the way we replace sodium and potassium back into their correct positions is through the use of sodium-potassium exchange pumps, which require ATP. So that's why the continuous propagation is energetically costly, is because we have to use a lot of sodium-potassium exchange pumps. So this is not very common. The more common type of propagation is saltatory propagation along a myelinated axon. So in the picture up top, you can see these things that kind of look like pink hamburger buns, right? Those are representing the myelinated areas of the axon. Don't forget, a myelin sheath is formed through neural glial cells called oligodendrocytes or Schwann cells. And basically, it's just a fatty layer that forms around the axon. Now, don't forget, the myelin sheath is not continuous along the axon. There are areas that are myelinated and areas that are not myelinated. So you can see there's little spots between the hamburger buns where there's no hamburger bun, right? So there's myelinated areas and unmyelinated areas. The myelinated areas are called internodes. The unmyelinated areas are called nodes, and they're also known as nodes of Ranvier. So what's going to happen during saltatory propagation is that the action potential is going to appear to jump from node to node, right? Saltare in Latin means to jump, so that's where this word comes from. We're going to have action potentials jump from node to node. The reason why this is happening is because we're not opening the voltage-gated ion channels where the internodes are, and that's because we don't have any voltage-gated ion channels at the internodes. We have layers of fat there instead. So we can only experience an action potential at the nodes. So we're going to experience an action potential at one node. Ions will travel through the axon underneath the internode, activating the action potential at the next node, and this keeps happening over and over again. Now you can see that we're going to be using way less voltage-gated ion channels, which makes this energetically much more favorable because we're displacing much less sodium and potassium. It's also much faster, and the reasons for that are actually pretty complicated, but suffice to say that this jumping from node to node actually allows the action potential to be conducted much more quickly along the axon. So let's check your learning. So pause the video here and see if you can answer these questions. When you're ready, play the video to see if you got the answers correct.